Good morning, student. I am Dr. Reeta Kumari, Associate Professor, D.A.V. PG College, Siwan. Today, my topic is protein structure and the classification. Now, proteins are large polymer of amino acids that are linked together by covalent peptide bonds. Proteins have complex structure and perform important function in all living organisms. Every protein has a unique three-dimensional structure, which is referred to as its native conformation and made up of only 20 different amino acids. Protein structure. Protein structure can be classified into four levels of organization. Number 1, primary structure, secondary structure, tertiary structure, and quaternary structure. Now this is the figure of different level of organization in protein. On the top, the figure is primary level of organization, second is the secondary, third is the tertiary structure, and the last the quaternary structure of the protein. Primary structure. The linear sequence of amino acids, residue, and location of disulfide base in a polypeptide chain constitute its primary structure. It refers to a specific sequence of amino acids. Primary structure is maintained by covalent peptide bond. Secondary structure. Regular folding and twisting of polypeptide chain brought about by hydrogen bonding is called secondary structure. Hydrogen bonding between hydrogen of NH and the oxygen of CO occur at stability of primary structure. Important type of secondary structures are alpha helix, beta pleated sheet, and beta bend or hairpin like structure. This is the figure of beta bend or the hair like structure. Now alpha helix. It is a spiral structure of polypeptide chain that is coiled around a longitudinal axis helical fashion. It is the most common secondary structure. It is produced by formation of hydrogen bond between peptide linkage 3 amino acids residue apart. The mean hydrogen bonds are formed between first and fourth peptide linkage. There are 3.6 amino acid residue in each turn of helix. Alpha helix. All hydrogen bonds are parallel, that is, in the same direction. The helix may be right-handed or the left-handed. Right-handed helix are more stable and it is the commonest secondary structure found in the protein. Proline is never found in alpha helix as it has amino group and not the amino group. As we know that the amino group present in the peptide chain or the protein, but proline has amino group. Hence, proline is never found in alpha helix. Glycine also tends to disrupt the helix. Example is keratin. This is the figure of alpha helical coil in the keratin. Beta pleated sheet. This polypeptide chain is fully extended rather than coiled. It is composed of two or more polypeptide chain. Two or more polypeptide chain running side by sides are stabilized by hydrogen bond. Glycine is the main amino acids here as it is produced kink. Proline is also found common and it may be parallel or the anti-parallel. Example is silk fiber. Now tertiary structure. It refers to overall folding pattern of polypeptide which form three-dimensional shape. It is maintained by weak non-covalent interaction like hydrogen bond, ionic bond, van der Waals force, etc. Covalent linkage play minor role in its formation. Example is myoglobin. This is the figure of myoglobin. Quaternary structure. Many proteins are made up of more than one polypeptide chain. Each polypeptide chain is known as protomer or subunits. The subunits are linked with each other by non-covalent bond. The structure formed by fusion of subunit is known as quaternary structure. Mainly, three bonds stabilize this structure: hydrogen bond, ionic bond, and the hydrophobic bonds. Example is creatine kinase, which is dimer in structure, hemoglobin, which is tetramer in structure, and immunoglobulin. This is a figure of hemoglobin. Now, different types of bonds which are present in the protein. Number 1 is the covalent bond, in which peptide bond and disulfide bond is present. And second is non-covalent bond, in which hydrogen bond and ionic bond is present. Primary structure, peptide bond present. In secondary structure, hydrogen bond is present, and in tertiary structure, hydrogen bond, ionic bond, van der Waals bond and the disulfide bonds are present. This is the figure of different types of bonds present in the protein. Now, classification of protein. Proteins are classified on the base of shape. Number 1 is the fibrous protein. The polypeptide chain extended along a longitudinal axis without any sharp bending, giving rod or needle-like structure which provide the elongated shape. The most of the structural proteins are the fibrous protein. Example is collagen fiber, elastin fiber and the keratin fiber. Second is the globular protein. The polypeptide chains are tightly folded and packed into compact structure like a spherical or the oval shape. Most of the enzymes transport protein like albumin and globulin, hemoglobin, myoglobin, antibodies, hormones are globular protein. Based on the physical and the chemical properties, proteins may be simple protein, conjugated protein and the derived protein. Simple protein. It comprises solely of amino acids, not other chemical groups is present in them. Example is albumin, globulin, protamides, etc. Conjugated protein. Conjugated proteins are the combination of protein with a non-protein part called prosthetic group and the protein part called apoprotein. They are divided into following groups. Glycoprotein, protein combined with carbohydrate is an example of glycoprotein which is present in the blood group antigen, immunoglobulin and mucin of saliva. Second is the lipoprotein. Protein along with lipid is the example of lipoprotein. Example is low density lipid or very low density lipid. Nucleoprotein. Protein attached with nucleic acids. Example is stone. Now phosphoprotein contain phosphorus. Example is casein of milk, vitellin of egg yolk. Fifth is the chromoprotein. Protein with colored prosthetic group. Example is hemoglobin, flavoprotein, visual purple, etc. And sixth is the metalloprotein, contain metal ion such as cytochrome and tyrosinase. Derived protein. Derived proteins are the degradation product of native protein. Progressive hydrolysis of protein result in the smaller and smaller chain formation. Proteones and metaproteins are primary derived protein. Proteases, peptones, peptides are the secondary derived protein. Thank you.